I think we should get started. Thank you very much for joining us today. I'm Scott Garrison. I'm what's called the Principal Investigator for the INRange trial that you all participated in. You don't know me personally. I am a family physician, and for the first 20 years of my career, I was essentially the same kind of family doctor that you all have. I was practicing in Richmond, BC at the time. I'm also very passionate about answering some of the questions around how to make better use of the therapies that are already in existence. Enough so that I went back to UBC to get training to do some of the research that I thought was missing, which ultimately led me to my current role, which is as a family physician researcher here in Edmonton at the University of Alberta. So you can see I have to be pretty passionate about research to move from Vancouver, which is nice and warm, to Edmonton, which is extremely cold, but also wonderful in lots of other ways. But really today is more about you. So my purpose in doing this webinar today was to say thank you for your participation, to let you know how much your contribution actually managed to achieve, because it goes far beyond just the trial that you participated in. And so my goal today is to give you a little sense of that, as well as let you know the results of the trial, which were in the card that we sent you. So I know you know the results of the question we were asking, but you don't fully know all of the surrounding things that the trial accomplished. So what's normal in an academic presentation, which this sort of is what's called the disclosure slide. So everybody has biases. I obviously have my own biases, but it's important for you to know whether the person talking to you has any financial interest in what they're talking about. So I have no relationships of any kind with anybody that markets any sorts of products or devices. My income comes entirely from my clinical practice as a family physician and from my academic salary at the University of Alberta. The grants that I do have for research all come from government sources, none from industry. And the same is essentially true for the study that you participated in. So the funders for the INRange trial that you were participants in were the Vancouver Coastal Health Research Institute. So that's a branch of the Vancouver Coastal Health Authority, so government. They provided some in-kind research assistant support for the trial. ENACT, which stands for Enhancing Alberta Primary Care Research Networks, also government funded. They provided part-time research coordinator support for the trial. And the Richmond Hospital Foundation donated $20,000 to cover things like the letters that we're mailing out and other sort of costs for the trial. So what I'm going to talk about is actually the history of the project, because it goes beyond warfarin. I'm going to talk about the trial you participated in and its findings, and I'm going to talk about what we managed to accomplish from carrying the trial out. And there'll be plenty of time for questions at the end. My talk time, I'm estimating, is going to be around 35 minutes. And at the moment, you're not able to share your camera or or talk by microphone. But you will be, once I finish the presentation, we'll open it up. And if you want to share your video or your voice, you're welcome to. Otherwise, you can ask questions by typing them into the chat, which is on the bottom right. And feel free to do that as we go as well. I'm probably only going to address the questions at the end, but if you type your question in as it comes to you, it'll be first in the queue when we get to questions. So what is this all about? What is what I'm trying to do all about? Well, it really has to do with becoming a learning healthcare system. So one thing that you may or may not be aware of is that there is data that's being captured all the time just through routine care. Things that you and I would consider to be very important, things we would want to prevent, like if someone has a heart attack or a stroke, those things are recorded in a database by the government. Those things are recorded largely for them to keep track of how they're funding the healthcare system. But all of the things that you and I would probably find most important to measure are already routinely being collected. If we could simply randomize some very simple things about how we use different treatments, we could really learn how to make those treatments more effectively and very inexpensively. And so this was about trying to do that. You're probably surprised to know that we don't already do that. There is research that happens in the system using that data, but it's more observational. And if you really want to learn if what you're doing is better than what you were doing before, you really have to randomize it. So an example for why you can't just use observational data to learn what's better would be, if I look back to the early days of my career, I remember going to lectures where we were told we should be offering estrogen to women when they became postmenopausal, flushing and so forth, they would use it for. But we were also told that it would reduce heart attacks and strokes. And the reason that we were told that was that if you just looked at people according to what they chose to do, whether they took hormones or they didn't take hormones, the people who chose to take hormones had fewer heart attacks and strokes. But when the randomized trial was finally done, where people were randomly assigned to take hormones or not take hormones, we ended up finding that it was the exact opposite. You were substantially more likely to have a heart attack or stroke if you were on estrogen, even though the observational data had suggested the reverse. And that's because there were other things that those people choosing to take hormones were probably doing that was giving them a lower risk. They may have been less likely to smoke. They may have been more likely to exercise, more likely to do other kinds of healthy behaviors. They might have been more affluent. There is an association between better outcomes and income. So we really do need to randomize things. So this was about finding a simple way to do that. And it was about trying to do it in primary care, trying to do it in your family doctor's office. The reason why it was important to do it in the community, to do it in primary care, is that almost all research that is currently taking place, most of which is industry funded, takes place in either acute care settings, like in a hospital, or in a specialty setting, so in a specialist office. And the kinds of trials that get done have inclusion and exclusion criteria that end up making only a very narrow segment of the population eligible for the trial. So it tends to be a lot of middle-aged white men, which I'm one now. But a lot of those trials exclude people who are older, exclude people who have a lot of different, we call it comorbidities, but a lot of different problems, medical problems that they're dealing with. Women traditionally were underrepresented in trials. And the real world is very different than that. The real world is people who have all kinds of different medical conditions, not just the one that you're studying. And it's very reasonable to question whether the risk and benefit of a new medication is meaningfully different between the person who studied, who only has maybe the condition you're studying and maybe only one other extra medication, and the person who's ultimately going to be offered this therapy who may have 5 or 6 medications, 3 or 4 other problems. are the risks and benefits different? So doing a lot of these studies in primary care is really important. So around the time that I was finishing my PhD work at UBC, I had the opportunity to talk at a large conference for family physicians in Vancouver. And at that conference, I pitched to them would they participate in a system like this if I built it? And so essentially I was, at that time I was talking about something which is probably still the thing I'm most passionate about, which is I think older adults use more medication than may be needed. A lot of family physicians believe that. And what I said to them was, think how easily we could do a study that answers the question of should we use less medication in older adults. You, the family physician, would use your electronic medical record to find everybody who's, say, over the age of 80 on 6 or more medications. You would scratch off the names of anybody that you think would be ineligible for the trial if they weren't able to consent, or might have some illness that would make it inappropriate. And you would tell us how many people were in your practice like that. We would send you the same number of envelopes, inside which is a letter of introduction about the trial and some information about the trial. You, the family doctor, would attach mailing labels. So the researchers have no idea who's being invited, only the family doctor knows. And the family doctor would mail them. If people were interested, they would see our number on the information package they would receive. We would, they could call us, we would tell them about the trial. We would randomize them to either have a visit with their family doctor to start trying to reduce their meds as much as possible, or to continue what they're doing. And then some years later, we would use the administrative claims data, which is already being routinely collected, to learn who was being admitted to hospital, who was losing their independence and being admitted to a nursing home, who was having a heart attack or a stroke. And we would hopefully learn if that made a difference. So at that meeting, so there were about 350 family physicians in the audience. 46 of them gave me their email address saying that they would participate in that if we could make it happen. So I was quite encouraged by that. I was still in BC at the time, managed to get some funding from the BC divisions of family practice. We created that logo that you see, we created the name, and I went around the province talking at different events and doing the same thing, recruiting people for a network to do that kind of work. And ultimately, when I got hired on at U of A, I've been doing the same thing in Alberta and at other events if I talk outside of those provinces. So then the question became, okay, well, what should we study? We wanted it to be something as simple as possible in the beginning, because we wanted to make it easy for the physicians because they were doing this for the first time. Virtually none of them had participated in research before. They were all doing this as volunteers. The docs were not paid a cent for anything that they do related to this. They're donating their time. So we wanted a very simple thing. So one thing that might be important is medication timing. So there are studies that have been done in rats, and subsequently a different kind of study that has been done in humans that show that there is a circadian rhythm to all of the organs in your body. So you think of sleep, you go through a period of time where you're asleep or you're awake. That's the main thing that you'll notice. But your body is governed by the DNA inside of your cells and The way that your DNA is brought into action is by individual genes are either expressed and in use or they're not. They're sort of quiet. And that can be measured by what's called messenger RNA in the cell. It tells you if that gene is being used. So if you do a study in rats where you look at different rat organs, you'll find that almost half of the genes, proteins that your body makes, have this 24-hour cycle where there are rush hours of expression of these proteins, expression of these genes, that are typically around dawn or dusk. And for each gene, it actually varies depending on the organ. So you might have one gene which is particularly active in the heart in the morning, but that same gene has its activity in the evening in the liver, right? Like it's quite variable. It's very organ specific. But so we know from a experimental sort of laboratory level that physiology is like this, and might it matter to humans? So there's one study that I know of in which it seems to matter. There was a study that was done on a particular type of chemotherapy where it turned out if you gave it in a way that gave more of the dosing overnight, that the toxicity was less, that people tolerated the chemotherapy better. But other than that, there was very little research that was being done in this area. There were some medications where, you know, your doctor would tell you what time a day you should take it. Like if you have esophageal reflux, you'll commonly be told to take H2 blockers. So there are things like ranitidine at bedtime, whereas with another kind of medicine called a proton pump inhibitor, you're told to take those in the morning. And there was a rationale for all that, but there is no randomized control evidence to tell us really if it makes a difference. So the one thing which was that was out there at the time was one study that was published in 2010 in Spain about the time of day that you would use blood pressure medicine if you had high blood pressure. And that study had suggested that there was a 61% reduction in a composite outcome that cobbled together death for any cause, stroke, heart attack, some other cardiovascular things. They added them all together, and they found that the group that took their blood pressure medicine at bedtime had 61% fewer heart attacks and strokes and deaths, like all those things together. It never changed practice because a couple of reasons. One is it was published in an obscure journal, which is not a mainstream journal, didn't get any media coverage, and also seemed unusual. Like if you had this groundbreaking trial, why would you publish it in an obscure journal? Why would you not publish it in a mainstream journal, which would likely take it? And it also sounded too good to be true. So, you know, when we treat blood pressure, like taking blood pressure pills versus not taking any, you expect about a 35% reduction for a typical person in those events. Same thing if you take a statin versus not taking a statin, you get about a 35% reduction. So is it believable that you would get a 61% reduction just from on top of the benefit of already taking the blood pressure pill, a 61% reduction just by what time of day you took the blood pressure pill? So there was a lot of skepticism about whether this was true. And we thought, okay, well, this is great. This is an easy thing for us to try to either validate or refute. And so we went about creating what we called the BedMed Study. So BedMed, very much like the study I just talked to you about, the hypothetical one for reducing medications in older adults. And the docs would use their medical records to find people who had high blood pressure and used blood pressure medicine. They would tell us how many letters they needed. We'd give them number of letters. They would mail them. If people were interested in participating, they would get randomized to take their blood pressure meds at bedtime or in the morning. And we'd look at the admin data to find out who had heart attacks, who had strokes, and so forth over time. So we applied for funding for that here in Alberta. There was a local competition, but they said no. And why they said no was what was important. And they ended up saying that, yeah, we think this is really important. We think that it's a great idea, but we don't believe that the family physicians will participate in the numbers that you say that they will, because research, traditionally randomized trials, are not done in primary care. Again, they're all done in specialty settings. So they didn't think that we could do it, which is why they turned us down. So that meant that we needed to do a study which was similar, a study that had to do with medication timing. And we wanted to learn the answer about whether the timing mattered, but we also wanted to demonstrate to the funders that, no, the docs would do this. And so that's what INRange was about. So the study question was, does the time of day that you use warfarin make a difference to its effectiveness as an anticoagulant, right? So morning use versus traditional evening use. Historically, most people were taking warfarin in the evening, and that was presumably because you're going to get your blood test done usually earlier on in the day. The doc gets the test result in the afternoon. They call you towards the end of the afternoon. If the time of day you take it is in the early evening, that's the shortest amount of time in between learning you need to make a change and being able to make a change. So it made sense. But there were other things that potentially could have affected it. Warfarin, the drug, has a very long half-life, and that means it takes quite a while for it to leave your body. So you can tend to get fairly stable blood levels if you had some way of actually measuring warfarin. But the liver is where it works, and the liver would see fluctuating levels. Whenever you eat anything that your body is digesting, it all gets brought to the liver first, because your liver needs to detoxify it. That's the liver's job. And so even though the blood would have sort of fairly stable levels of warfarin, the liver would see large amounts of warfarin around the time that you ingest it, falling to some baseline level and then always going up every time you ingest it. It's called the first pass effect. And also the way that it worked could have mattered, because even though warfarin has a long half-life, the way warfarin works is your liver is making clotting factors to stop you from bleeding that it releases into the blood. And in order to finish off those clotting factors, the liver needs vitamin K. It's part of the process. It's one of the tools to finish off the clotting factors. Warfarin stops your body from being able to use vitamin K effectively, or it slows it down, which slows down the production of the clotting factors. But even though warfarin had this long half-life, vitamin K has an ultra short half-life. Vitamin K is only, the half-life is 2.5 hours. So it's in and out, fairly quickly. And it's also in fairly select foods. It's in sort of green leafy vegetables. And very few people, at least not me, takes green leafy vegetables in the morning. Probably don't even really have that much at noon. It tends to be the evening. So, and it can be quite variable. So vitamin K intake could be highly variable. It would tend to not be in the morning. Could it be better to take the warfarin in the morning when the vitamin K level in the liver would be consistent, lower, but consistent? anyhow, we had speculated that maybe morning would be better. We really didn't know, but nobody knew what time of day would be better. And so that's why we began the study. The main reason why we looked at warfarin as compared to any other medication was that warfarin has a blood test that's very predictive of its ability to prevent heart attack and strokes. We would have loved to have done a trial that looked at counting up the number of heart attacks and strokes that were, or blood clots in the legs and moving to lung, which is called pulmonary embolus. We would have been loving to look at those important things, but you would have needed thousands of people in order to show a difference. Whereas if you look at the blood test, which you can measure on everyone, you can answer your question with a much smaller number of people. And we had to run this trial on a shoestring budget. So what you're seeing here is a graph that shows you the risk of the bad things that we don't want to have, depending on what the INR level is. And there's two curves there. I don't know if you can see my cursor if I move this over the screen. Hopefully you can. The dark line here is the risk of having a clotting consequence. So the things we're trying to prevent, right? So blood clots in the legs that might break off and go to the lung, or having a stroke if someone has some reason why a clot could form in the heart. So this is the risk of those things happening. And the dotted line is the risk of the adverse consequences, the side effects of taking warfarin, which is bleeding. This one doesn't include all bleeding. Not all bleeding complications are equivalent. It's much easier to deal with somebody who's bleeding in their bowel than it is if you bleed into your head. That's the worst thing that could happen. And so this here is showing the risk of a bleed into your head. So, but you can see that there's this range of an INR between around 1.8 and 3.5, where you're at the lowest risk of all the bad things happening. And so one thing that we can look at is the proportion of time that people spend in the therapeutic range of the blood test. It's called time in therapeutic range. It's abbreviated TTR. So you'll see that in the slides that are coming up. So this is a very busy slide, intended more for an academic audience, but this just talks about the methods of the study. So your family doctor that was participating in the trial probably had seen me talking at one of those conferences and had given me their email address there, or they could have been an office partner of somebody who had been there and they decided to participate as a whole office. But they know who uses warfarin. Most of them maintain sort of a flow sheet, so it's very easy for them to tell who in their practice is using warfarin. So they told us how many people in their practice were using warfarin. Again, we prepared envelopes for them, inside of which is a letter of introduction. Everybody had the same letter of introduction, although the physician probably would have signed it themselves, but they had sort of approved that, yes, this is a reasonable letter. And then an information package about the trial. So they applied the mailing label. So we have no idea who was invited, but clearly you were one of them. And then if you wanted to participate, you would have called the number that was on the line there. You would have been speaking with a research assistant who would have been out of Vancouver Coastal Health Research Institute. Initially, we would have been wanting to make sure you were eligible for the trial. So you had to be living in your own home or in assisted living, but not, if someone was in a nursing home, they couldn't have controlled their medication timing well. So that's why they were excluded. You needed to be using warfarin in the evening. You had to be expecting to use it long term. didn't matter why you were using it. And we had to receive, once you had said you wanted to participate, we had to receive from your doctor INR readings, right? The blood test readings. We asked for the last 6 months. Ideally, we needed to get at least 3 months worth of it, the most recent 3 months. And there had to be at least 4 INR values that we could use that were in there. And then you were randomized to either take it in the morning or to continue using it in the evening. And then the things that we looked at, so we, the study followed you or your INR values, which we got from your family doctor at the end of the study. They faxed them back to us 7 months after you were randomized. And we compared the proportion of time that you were in range of the blood test in the last 6 months. So the first month, we didn't count it in case there were some changes that might have happened as a result of changing the timing. And we compared that to the 6 months before you were randomized. And looking at our primary outcome, which was percent change in the amount of time you were out of range, so which we hoped would reduce. We were also looking at the absolute values of the time in therapeutic range itself. We were looking at the percentage of people who had great control, which meant their TTR, their time in therapeutic range, was better than 75%. We looked at the percentage of people who had poor control, which was the time in range less than 60%. We looked at the maximum out of range value, the minimum out of range value, the percentage of time that you were above the range, the percentage of time below the range. And we did look at whether people had cardiovascular events, heart attacks, strokes, blood clots, bleeds, when they were in the study. But we knew from the beginning we weren't going to have the power to answer the question about whether that was any different. So we were very successful at engaging the family physicians. 236 family physicians were recruiting their warfarin-using patients across 54 different communities in Western Canada. You can see the communities there. Some of them that are nearby each other would be aggregated. So Richmond would be aggregated with Burnaby, for instance, or St. Albert with Edmonton. But you can see the distribution on the map. Of the people who received a letter, 16.7% of them called to enroll in the study, which is great. And so there were 351 people who we spoke to assess eligibility. We excluded 134 of them. The main reason, like 93 of those 134 were excluded because they were using warfarin in the morning already, which was surprising that there was that many, but that was the main reason for exclusion. For 10 people, we couldn't get the INR values from the doctor, or what the INR values we did get were insufficient for us to enroll them in the study. Too few, for instance. And 10 people, once they mulled it over, didn't feel comfortable potentially having to change the time. So 217 people were randomized. And as far as following them, if you compare this to other randomized trials, it was excellent as far as there's only 3.7% of people who either didn't do what we'd asked them to do, like they ended up taking it at a different time, or for some reason we weren't able to continue to contact them to get information. So 3.7% is really low compared to other randomized trials. And that was in the intervention group where we were asking people to make a change. In the control group where you were just continuing on with whatever you were doing, it was much lower, 1 point something percent. So who was in the study? So 109 people in the morning group, 108 in the evening group. You were about 50-50, male, female, randomly, there was a bit more males in the morning group. The average age was around 73 years. And the reasons for using the warfarin, about 60% of you were using it because you had atrial fibrillation. About a quarter of you were using it because you had a blood clot in your leg, called a DVT, deep venous thrombosis, or a blood clot in your leg that broke off and went to your lung, pulmonary embolus. And about 10% had an artificial heart valve. Again, clots can form on that and could lead to strokes, which is why people use warfarin for that. And then about another 10% or so used it for other reasons, and which were quite variable. So they might have had an aneurysm, there's a sort of bulging area on their heart, or they had a stroke and nobody could figure out why they had the stroke, and they still put them on warfarin, like that kind of thing. Fits in the other category. In addition to warfarin, on average, participants used 5 other medications, prescription medications. So warfarin was the 6th. And on average used 2 to 3 vitamins or herbs, some sort of supplement. The target ranges, so for about 90% of people, your doctor was shooting for a range between 2 and 3. For about 7 or 8% or so, they were shooting for between 2.5 and 3.5. That's normally the people who have mechanical heart valves, which are the target range is that higher end of the range. And a small number of people in the morning group, 5 of them, had other ranges. And that typically would be, there might be some doc who was shooting for between 2 and 3.5, or the odd one had a very narrow range, like they wanted it between 2.5 and 3, which is hard to achieve. But the time in the baseline before we did anything, the time in therapeutic range was around 72%, right? So the participants were in range at baseline about 72% of the time. So what did the study find? Well, we've sent you the postcard. You already know what the study found, which is that it really makes absolutely no difference what time of day you take it. So again, this is a busy slide intended for an academic audience, but if you look at the very top one where it says absolute change in TTR. So if you remember, I said that people were in range 72% of the time at baseline. So in the morning group, they improved by 2.9%. So they went from 72% to 74.9%, essentially. And in the evening group, they went from 72% to 75%, right? So you can see that those numbers are essentially exactly the same. And then our primary outcome was the percent change in the proportion of time that you're outside of range. And that was 4.4% different, which is not really different. It's just, I mean, the numbers are not going to fall exactly on, like that small amount of difference is not statistically significant. It's just likely to be random, minor difference. So the percentage of people that had good control was essentially the same in both groups, 56% in the morning, 54.6% in the evening. The percentage of people who had poor control, again, the same, 22% in the morning, 22.2% in the evening. Sorry, I hope I said poor control. And the maximum and minimum out of range values were essentially the same. Percentage of time you were above range or below range, they really weren't any different between the two groups. Which led to our conclusion that people who use warfarin should take it whenever it's most convenient for them to do so or easiest to remember. So as far as what has stemmed from the study, there's two main things. So our results were published just before COVID epidemic hit, so in January 2020. And of course, publishing is great, but you need to get it in the hands of people that would see it. So I would wager that probably all of your doctors, the majority of your doctors, probably have a web service, a paid web service that they paid for, called UpToDate, which they use in the clinic. And so this, if something happened that's beyond the norm for them, and they need to look something up, they'll usually go to this website, which is called UpToDate, and we'll be looking for it in there. So very shortly after our study was published, UpToDate revised. They didn't do it for us; they revised periodically, but they revised their section on warfarin anticoagulation to cite our study and to say that it probably makes no difference what time of day you use it, which is great. So little by little, that will probably change. But the main thing that happened as a result of INRange is before the trial was even published, we were able to show that because the physicians and because you participated in such large numbers, right, like 236 docs and sort of 16% of people receiving a letter choosing to participate. When we were able to show that for the next grant opportunity that came up, we got the grant. So Alberta Innovates ended up funding the BedMed Study. We started that, it's been a few years ago now. The opportunity came pretty much just as we were able to have final data for the INRange trial. That the funding from Alberta Innovates, we were able to match with Canadian Institutes of Health Research to actually do the same study in multiple provinces. So BedMed is now ongoing in 5 provinces, in BC, in Alberta, in Saskatchewan, in Manitoba, and just now we're ending with recruitment in Ontario. There are somewhere around 370 or so-ish family docs that are participating. There will be more as the Ontario docs join in. And there are around 3,000, I forget the exact number now. It's somewhere around the 3,800-ish number of participants who have had their blood pressure medicine randomized to morning or bedtime. So we will be able to, that study will end, the data will be mature at the end of 2022, and we'll know the answer towards the end of March, probably, in 2023. And then, of course, we have to have a conference or some way in which we can release it and have it peer-reviewed. But so I also want to end again by saying thank you. If you hadn't participated in INRange, we would not have been able to answer the question about warfarin, but maybe even more importantly, we would not have been able to carry out or get funding for the BedMed study or to build this whole platform of research, which we're moving on. Like my next trial is going to be trying to do the one where we look at minimizing medications in older adults. But that whole program of research wouldn't have been possible if INRange had not succeeded, and you really made it that. The other thing I would say to you is, I don't think we necessarily did it so much in the INRange trial, but in the BedMed study, we've been asking people why they choose to participate with a whole bunch of different options. And almost invariably, the people that are participating, are doing it because you're trying to help someone outside of yourself. You're trying to help change the world in meaningful ways. And I just think that's wonderful. I think all of you are giving altruistic people that are really making a difference. And I wanted to make sure that you could see how you did make a difference, both for this study, but also for all the studies that we're going to be able to hopefully do as a result of it. Like I have a young doctor who was a grad student of mine now, who's interested in doing trials, and she's starting to build her own trial in it's in a nursing home population, right? But still, a family physician building a trial in the community. These are all new things, and so sort of paying it forward. So thank you for helping me do this, and I will be helping other students do other projects leading on from it.